So here's that again. I keep on showing the same picture. This is that chemical reaction going on where you're taking CO2, H2O. So what you want to notice here is all this stuff, right, is going to be happening in an aqueous environment in your body, right? All these dissolved water in the blood, or interstitial fluid, or intercellular fluid, as well as, the blood plasma itself, the interstitial fluid. Everything we're talking about here is happening in water, right? So water is the background of everything we're looking at here, and it's going to be a big player in these reactions. So let's look at that a little bit, and the importance of water, right, in all living things, right? This is a mummy, right? This has been dehydrated. Basically, this is what you'd look like kind of like a dried raisin if all the water was taken out of you, right? So it's about 60% roughly of all your body weight. And in any individual cell, it's about 80% of the cell weight, right? And again, the key to water's properties is the fact that it's a polar molecule attracted to itself through those hydrogen bonds right there. So water is a key point right here. Beyond that polarity stuff, or maybe part of it really, it's going to be part of the chemical reaction itself, right? So this reaction where I talked about the breakdown of sucrose into glucose and fructose by the addition of a water molecule. These are hydrolysis, right? Hydro means water, lysis means breaking apart. You're basically adding this water molecule over here and then breaking these two apart right here. So you get glucose and fructose. And then the opposite reaction, dehydration, right, is going to actually remove that water and form this chemical bond in between these to make sucrose. Right? So water is involved directly in these. So this will be true for peptide bonds as well. Peptide synthesis, joining of amino acids. It's going to see that same hydrolysis and dehydration synthesis. And then the other major part we talked about yesterday is those fluid compartments, right? We have water, intracellular water, interstitial fluid, part of the extracellular fluid, all the fluid in the plasma, right? Everything that is within, the blood, the fluid here is all dissolved in water or part of the water, right? So, when we look at the difference in between the outside and the inside, right? All this stuff, all these ions and molecules and stuff are dissolved in the water, right? On either side of the cell right there, right? So it's a solvent, things are dissolved in it, right? So when you take some salt crystals, starting with this big one right here, even though the ionic bonds, right, are stronger than the polar bonds, you have a whole bunch of them, mass numbers of them go around, they start to attack it and start pulling away sodium ions, pulling away the chlorine to form these little hydration cells, right, to break down those molecules so that these ions are going to exist by themselves as ions in the solution, right, totally interspersed. And again, water can also dissolve non-ionic bonds like covalent molecules through these hydrogen bonds, right, of the molecule itself, if it's a polar molecule. So these little OH groups on a glucose are going to allow these glucose molecules to be, one of the main components. That's what blood sugar is, right? So ionic dissolution or sugar or covalent dissolution, right, it's a little different because here the actual atoms or ions are being broken up. Here the molecules are just separating from one another, right? But they separate them out. And it's the same sort of idea, right? That they're dissolving in the water and they're going to basically become part of the solution. Here's a sugar molecule, sucrose, right? See all these little hydroxyl groups are. Each one of these can interact with a water molecule right here. So that's what's going to separate the individual little the sucrose molecules from each other, from a crystal. So when that happens, right, these are the terminologies we're going to use for these things that are dissolving in water, right? Sugar, in that case, was the solute, right, the solid particle, and it's going in water, which will always be, in this class, always the solvent, right? If you're taking chemistry, you might talk about organic solvents that will dissolve non-polar stuff, but for us, our purposes, solvent will always mean water, and we'll just call it water, right? So the sugar would be dissolved in the water, right? Salt, same thing. You're going to take the solute, put it in water, and it's going to break down. So it's all evenly dispersed throughout the water, right? Such that you wouldn't be able to tell, like, until you start drinking it, whether it's sweet or salty or something, whether that was actually dissolved in there, right? Because completely dissolved in there. So what this gets at is this idea of different types of mixtures, right? And what we just talked about here was a solution, evenly mixed small solutes, and then the sort of fluid that that solution would make, if it was a pure solution, would be transparent, right? If there's a little bit of bigger solute particles, they're still going to be evenly mixed, right? They're still in solution, but they're bigger, but they're big enough so that if you shined a light through it, they'd be translucent or opaque, not completely transparent, like milk, right? This is called a colloid. Right over here, bigger particles that they're evenly spaced, right? So between these two, this one is just like milk versus salt water or sugar water, right? And then the next one is called a suspension. Large particles are mixed in water as long as things were moving around and stuff, but if you let it sit still or you spin it down, those would settle down because they're too big to really stay in dissolved and mixed up evenly right there. So these are called suspensions. So there's three different types of mixtures, and let's talk about why this is important, right? So blood is kind of a mixture of all three itself, right? The blood itself is a suspension because of these big cells here, but there's also stuff dissolved in here. All salt is dissolved in here, glucose is dissolved in here. Then you also got bigger proteins, right? So the plasma itself is really a colloid solution, but a colloid, but there's also, you know, dissolved molecules in there. So it's a pretty complicated one. So if you took this blood, right, you spun it down, you'd separate all those large particles, right? This is what makes it a suspension. So you take that, spin it down, and then on the top layer right here, the lighter stuff here, this would still have all the dissolved components, right? If you spun it down a lot, you probably get the bigger proteins to get in there as well, right? But you're going to separate that based on the weight, the size of these particles, in this case, the red blood and white blood cells, right? So just to go on that a little further here, you don't have to know any of the details here. Just know, like, when we're talking about these solutes, right, in the blood, right, you're talking about electrolytes, meaning sodium, potassium, calcium, all that stuff within the blood, sodium bicarb floating around the blood. There's glucose in the blood, there's oxygen and carbon dioxide, right? There's hormones, there's other waste products floating around the blood, right? Either as a solution or there's bigger blood proteins, right? The albumin is a big one, right? These are also floating around the blood right there, right? So these are different types of solutes. And here's the important point I'd like to make about that, right? Here's a capillary, right? And blood is being forced through from the blood pressure. Over here, water, oxygen, glucose and stuff is coming out of it based on this pressure. And you also have all these bigger proteins which don't leave over here. And so these bigger proteins that stay in here are going to have a large effect on what happens over here, right? So here's another picture without the actual stuff right here, but you see this term right here, colloid osmotic pressure. This is all the proteins over here. This has a certain pressure. There's a certain amount in the interstitial fluid, and there's a certain amount of colloids over here. And when we learn about osmosis and diffusion and stuff, we'll see that water is going to tend toward this. It's going to move toward this greater concentration of proteins, right? So you'll see this term, colloid osmotic pressure. Often that'll be referred to those big proteins in there and stuff that are creating this osmotic pressure. Again, we'll talk about this in the next unit when we talk about water and diffusion and stuff, but that's just a key point about these mixtures right here. All right, so that was these mixtures, right? They were dissolved in there as solutes or whatever. But the other point about this blood here is, this, when you look at the makeup of blood, right, 59%, 55% plasma, 45% formed elements like white blood cells, red blood cells, platelets. But for here, you're looking at this percentage-wise, right? 7% proteins, and then you could break down these as a matter of how much it makes up the proteins. Like albumin is most for proteins. Over here, they're not giving it, but overall, it's 1.5%. And so these are the concentrations, right? Which brings us into our next sort of topic. And so when you are a nurse, most people here becoming nurses, you'll have a little cheat sheet like this when you're given these IV solutions. And you might have glucose ones over here where you're going to say give them a D5W, which will be 50 grams per liter of glucose, or maybe a D10W, which is going to be 100 liters, twice as much glucose over here. And then you'll know that one of these is isotonic, that is pretty much matched with the blood, which again, we'll talk about later. Or one of them has more sugar than the blood, and referred to as hypertonic, right? So these would be major things to consider when you're sticking, fluid in people's arms, right? Or it could be a salt solution. If this was the isotonic one, which is about the same concentration as your blood, or you could put less salt that's in your blood and it would be a hypotonic solution, right? Each of all these have particular purposes, right? Percent saline is going to be, relative to the solution here, and then it's going to be hyper or isotonic or hypotonic, right? So you could label that as maybe 0.9% saline, or you could also call it 154 millimoles per liter of sodium and chloride, right? These are two different ways of saying the same thing, right? This is different ways of expressing concentration. You could do it in terms of percent, that is mass per volume. If you want to make a 10% sodium chloride solution, you take 10 grams and add it to 100 millimoles. This is just the standard of how we define this percent. So this is one way of looking at it, or you could look at it in terms of what's called molarity. You take 1 mole, molar solution, and to make 1 molar solution, you're going to dissolve 1 mole of sodium chloride, which happens to be a particular weight, and you put in enough water to make 1 liter, right? That'll be a 1 m solution of sodium chloride. So what this means, this mole thing, if you don't remember this from chemistry, it's just a number, right? And so you know what a dozen is. If I told you how much does a dozen eggs weigh, eggs weigh, you would say, I don't know, like a pound or something. I don't know how much a dozen eggs weigh. If I asked you how much a dozen bowling balls weigh, you'd know it's a lot more, right? So they're both the same number, but because they're different sizes, they're going to have different weights, right? They're going to have different densities and stuff. So 1 mole of sucrose has a much greater volume than mercury, because mercury oxide is much denser, right? And you can fit a lot more. But this has the same exact number as this of molecules. Think of that dozen marbles versus bowling balls or whatever, right? Same number, but it's going to have a different volume and weight, basically. Right? So you're going to take, if you want to make a 1 m of sucrose, you're going to take this much, pour it in a mixture, and you'll have a 1 m solution. If you want to make a 1 m mercury oxide, you take a lot less, pour it in, you'll still have a 1 m solution. Right? So this is a different way, because sometimes it's really more important to know about the number of molecules in a solution. So this comes into play with that whole colloid osmotic pressure. Right over here, the number of actual particles is going to be the factor in whether, diffusion happens one way or another, right? The number of particles on the outside or the inside of the cell is important. You don't talk about percentage outside, you really talk about millimoles, right? The concentration of number of actual molecules, right? And then there's other terms like milliosmoles, right? Because sodium chloride breaks up into 2, so there's so-and-so, right? So this will be another important point, right? You use this measurement, this concentration, calling them the number of the concentration of millimoles in a millimole units, right? Or mole unit. Clear on that whole thing? Concentration, that'll come up quite a bit too when we look at the differences between concentrations, maybe intra or extracellularly.